Bismillahirrahmanirrahim. 9700 June 23 paper 4-2. This is the third video which contains questions 8, 9 and 10. We start with question number 8. A, in plants such as rice or rhizospira, rubisco has a low rate of activity which in turn affects the rate of photosynthesis. The cereal crop sorghum has a high rate of activity of rubisco. So rice has a lower, sorghum has a higher rate of activity of rubisco, which is used in the Calvin cycle. A genetically modified variety of rice was produced. Parts of the cottony structure of rubisco and rice were altered to be the same as the rubisco and sorghum. So basically we are altering, genetically modified, we are altering the rubisco which was present in rice to rubisco in the sorghum. Rubisco, you know, is an enzyme which catalyzes the fixation of CO2. So RUBP combines with CO2 in the presence of rubisco. The rate of activity of rubisco in non-GM rice and GM rice was measured at different concentrations of atmospheric carbon dioxide. So the rate of activity of rubisco in non-GM and GM rice. Now GM rice, what have we done is that we have altered the rubisco. So the results are shown. Now you can see the results here. Rate of activity of rubisco in the non-GM rice, you can see how the rate of activity is increasing. Let me just give it another color. So it's increasing till about 200 CO2 concentration, a little more, and then it is going to level off. But if you look at the, this is the non-GM rice, but if you look at the GM rice in which we have altered it, it goes up to, it is going up 200 and 200 atmospheric 4 is the rate of activity. It even goes up below above that, and it is something between 4 and 5. The activity, while the non-GM rice is only between 2, something between 2 and 3. So it's somewhere here, you see, and this is somewhere here. So here, so a little bit more than 4, and this is a little bit 2.5 or something like this. Compare the two curves in figure 8.1. Explain why the curve for non-GM rice levels off. So first is a compare, and then it is an explain. So this is the first is a compare, and then there is an explain as well why the curve for the non-GM rice levels off. Now this is for how many marks? This is for 3 marks. Now what can you do the comparison? Higher rate of activity in GM rice throughout. So let's look at the graph. So higher rate of activity in the GM rice. Higher rate of activity in the GM rice throughout. Low CO2 concentration, rate of activity for both increases. So this is the one, it increases here as well, and this also increases in this 0 to 200. Then you could give me comparative figures. Atmospheric carbon dioxide concentration at 0, the non-GM rice is 0.7, the GM rice is 0.80, or you can give it at 100, or even at 200. You could have said, you can see here at 200, you could have said that the non-GM rice is 2.4. So you just draw a line here and read off all these. So it's 2.4 and the non-GM rice, and it is 3.95. It is 3.95 here. Here it is 3.95 and here it is 2.4. So you can give me all these figures. They're given in the mark scheme. You could have given me at 300, at 400, at 500, at 600, and you just give me a comparative statement. Now, what are we going to do about the explanation? Now, the explanation, why does it level off? Because CO2 concentration is no longer limiting. At this point, when it levels off, let me just show you here. When it levels off here, I always say the x-axis, this is no longer limited. So CO2 concentration is no longer limited. Now light intensity is limiting, or you can say temperature is limiting, or you can say less RUBP available, or rubisco active sites are saturated. So explanation is that, you know, you have to give me something which is a little bit of biology. That at this point, when it levels off, because it said explain why the curve for non-GM rice, this is the non-GM rice, levels off. So it levels off because now carbon dioxide is no longer the limiting factor. Whatever is on the x-axis, that becomes no longer the limiting factor. Or you can say light intensity is limiting, or temperature is limiting, or rubisco active sites are saturated, or you can say less RUBP is available. So a lot of mark scheme points which you could have given me. So higher rate of activity throughout. In low CO2 concentration, rate of activity for both increases. You've got to see the similarities in the graph and of course the other points as well when you give me data figures from the graph. So at 200 milligram dm cube, GM rises 3.95 arbitrary units, and then it's non-GM rises 2.4. Explanation: CO2 concentration no longer limited when of course it has leveled off, and you could give me the other answers like light intensity limiting or temperature limiting. You couldn't give me any of those points as well. Then it says suggest which part of the disco molecules was altered to produce the GM variety of rice. The active site. Naturally, the active site would be altered to facilitate it to work better or to work slower anyway. Then coming into B part of the question, figure 8.2 shows an outline of the Calvin cycle. Name the process that involves the enzyme ribisco. What is that called? You can say carbon fixation. Carbon fixation. Then it says state the type of reaction that occurs when GP is converted to TP. GP is reduced to TP. That's what we say all the time. GP is reduced. So you could have said reduction. Reduction. Then it says RUBP is regenerated from molecules of TP. State how many molecules of RUBP are produced from 10 molecules of TP. Now you know RUBP is a 5 carbon, and 10 molecules of TP means 10 into 3, 30 carbon. So 30 divided by 5 would be 6. It is how many molecules of RBP are produced from 10 molecules of TP. But then you realize it could also be 5. Why? Because 3 exits. So TP minus 3 would mean less than 3 carbon exits. So you could have said 5 as well. 6 or 5 both were allowed. Then it says part 4, TP molecules that are not involved in the regeneration of R can be used in the synthesis of other molecules. State 2 molecules. Well, of course, you can say a whole lot of them. You can say hexose, glucose, you can say sucrose, you can say starch, you can say amino acids, you can say glycerol or fatty acids. So you can say a whole lot of things, but basically I would have said glucose or fructose is one possible answer. The other answer is you could have named a disaccharide, you could have said sucrose or maltose. You see how it's a category. Glucose and fructose is one marking point. Disaccharide is sucrose or maltose, and then you could have said starch, or you could have said cellulose, so that would be a polysaccharide. You see how the marking scheme is being made. Starch or cellulose, and then you could have said that maybe that's attached to a nitrate and then it results in an amino acid, or you could have said glycerol or fatty acid. So you see, you have to see how it is under different categories. First was a monosaccharide, then disaccharide, then polysaccharide, then the monomer of proteins, and then the monomers, or not the monomers, but the basic constituents of fats or lipids or triglycerides. And then coming to question number 9, interesting, the passage, I love these questions, fill in the blanks. They make life so easy. They make it so easy for us teachers to check the papers. The passage outlines the endocrine system. Complete the passage by using the most appropriate scientific terms. The endocrine system consists of tissues and glands, which secrete hormones such as insulin, glucagon, and ADH, antidiuretic hormone. Glucagon only affects target cells that have complementary receptors which are located on the cell surface membrane. On the cell surface membrane. Glucagon binds to these receptors, and this leads to the production of a second messenger, or you can see CAMP. cyclic AMP, or you could have said second messenger. Which transfers the signal throughout the cytoplasm. The target cells for insulin are in the liver, muscles, and adipose tissue. The target cells for ADH are those of the distal convoluted tubule and the collecting duct. Of the kidney nephron. These hormones are involved in homeostasis, which is the maintenance of a constant internal environment. Oh, very easy, 6 marks. I'm sure everybody could get that correct. That was an easy one. Then it says, of course, we come now to the B part of the question. The blood glucose concentration of a person was measured at regular intervals after the ingestion of a meal rich in glucose. Figure 9.1 shows the results of the investigation. Blood glucose concentration, AB, glucose-rich meal ingested. I think I could have a 3 milk cake. Blood glucose concentration in millimole dm cube. So we started it here, 0, and then you see it is peaking at 1 hour, and then going down, and then slight increase at 4 hours, and it goes down. So it says, let's look at the graph, let's look at the question. Explain how the blood glucose concentration is reduced during phase A of the curve. Phase A of the curve. Let's look at phase A of the curve. This is the phase A of the curve. Why has it got reduced? Well, of course it's got reduced. Why? Because insulin is produced, insulin increases the uptake of glucose into the cell, increased permeability of the cell membrane, which is the GLUT being added to the cell membrane, increases the respiration of glucose and stimulates glycogenesis or conversion of glucose to lipids. So simple, easy question. So very basic, insulin produced, increase in uptake of glucose into cells, GLUT added to cell membrane, that gives you the more details. Why would it increase the uptake of glucose? Then of course there's increased respiration of glucose, more ATP will be formed, and stimulates glycogenesis. What is glycogenesis? Glucose to glycogen is glycogenesis. Genesis means formation. Or you could have said conversion of glucose to lipids. So these are 3 marks. We had about 5 mark scheme points. So very easy to get these 3 marks. Then it says, part 2 of the question suggests why the blood glucose concentration increases again during phase B. Let's look at phase B. What is happening at phase B? Why is this increasing again? Because it went down. So why is it increasing again? You have to talk about glucagon. So glucagon being produced. That's why the glucose concentration is rising in phase B. So if you gave me anything about glucagon, well, you'd get the 1 mark for that. So glucagon is being produced, and that results, of course, in the glycogen being converted to glucose, and the glucose entering the blood, and of course the blood glucose level rising. Now coming to the last question. As you know, the essay questions have been abolished since the syllabus changed in 2022, so we don't have any of those questions anymore. Many zoos around the world are involved in captive breeding programs for endangered species. Outline the role of zoos in the conservation of endangered species other than captive breeding programs. So how can the zoo help in the conservation of endangered species? But we are not going to talk of the captive breeding. So what can the zoos do? Well, of course, the animals are kept away in a safe location, away from predators and hunters and poachers. Then we do research on them about their disease control or their diet or their behavior. Then in the zoos, we can have fundraising programs by which we raise funds to help in the conservation or to maintain their habitats or to prevent the destruction of their habitats. Then you work with the local government and help in the conservation projects. Then you educate and raise awareness among children, among people at large, and so that people don't hunt them and they're not poachers and all to kill these animals. And of course you work on their veterinary and health. And so that they suffer less. There are certain diseases which are rampant, and you can produce a vaccine or something so that they can be not suffering from those diseases. So outline the role of zoos other than captive breeding. And I'm sure everybody just wrote about captive breeding because you all don't read the questions. So safe location. Then at zoos we can do research on disease control and how to give them a good diet, or if they have any other diseases like worms, etc., then you can test their feces and check out. Then you raise funds. You can arrange people to come and do fundraisers. Then work with local governments, education, awareness, and exploring veterinary help which we can give them to prevent them becoming extinct. Now coming to the B part of the question, Chester Zoo in the United Kingdom controls the European captive breeding program for the eastern black rhinoceros, whatever. Figure 10.1 shows an eastern black rhinoceros. Wow, amazing. In the captive breeding program for the eastern black rhinoceros, 15.8% of females breed each year, while in the wild, 23.7% of females breed each year. Suggest reasons why fewer females breed in captivity. Why fewer females breed in captivity than in the wild? Unnatural environment. Maybe the effect of captivity is on the fertility. There's less ovulation, or there is some other pattern in their cycles is disrupted, then refuses to mate, then low availability of suitable mates. And then, of course, in captivity, there's stress, there's pressure. They're not in their natural habitat. They're enclosed in a small space, and zoos control the number they breed. So zoo capacity is also another factor. So one of these, you could have written 4 of these points, so you could get the 4 marks. So unnatural environment, captivity may reduce fertility, refuses to mate, low availability of a suitable mate, stress or pressure in a controlled environment. They are not used to this. They used to be in very, very spacious forests where they can just roam around, and here it's a very restricted place, and zoos control the numbers they breed. Now coming to the last part of the question, and that will finish this paper. Sometimes assisted reproduction is used in captive breeding programs for the eastern black rhinoceros. Figure 10.2 outlines the process of IVF that is used in these programs for hormone treatment. Then mature oocytes develop, oocytes collected, then A. What is A? Fertilization. What is the oocytes collected, and then embryos have been produced. So here what is done? Sperm or semen has been added. Or you could have said sperms added. You could have said that as well here. Then embryos produced, then implanted into original female, and then B, and then C. Outline the event A that occurs in the production of the embryos. So you could have said fertilization occurs or sperm added. And the 2 other possible outcomes B and C for the embryos that have been produced. So what could have happened to those embryos? Either we put them in the original female, of course, then they will develop into the fetus and then the baby. B and C could be surrogacy or cryopreservation. So this could be surrogacy, putting it into another animal which is not of another species, or surrogacy. And this could be cryopreservation, in which you preserve these in a very low temperatures. Cryopreservation. It's one word, of course. Let me write it here so that you know cryopreservation. So good question, and that was for 3 marks. 3 marks, one was for fertilization occurs, or if you said sperm with semen added. 1 mark is for surrogacy. The 3rd mark is for cryopreservation. and that is a total of 10 marks, and that was the last question, and that finishes your paper 4-2 for the June 2023 exam, which is the last exam that was held.